Good morning, Class 7th. This is your biology book, and I'm your teacher for the session, Sonali ma'am. Okay, so we have completed our first and second chapter in our previous videos. Okay, all the question answers, all the diagrams were discussed in your previous chapters, in your previous videos. Okay, now from today onwards, we are going to start our new chapter. That is your chapter number 3rd, and this will be the last chapter for your half yearly exam. Only 3 chapters are coming for your half yearly exam. That is chapter number 1, 2 and 3. 1 and 2 we have completed in our previous videos, and 3rd chapter we are going to start from today. It is a little bit lengthy chapter, a tough chapter, okay, and there are so many things to learn in this chapter. Okay, so listen attentively. If you are unable to understand anything, you can ask me again. Okay, now kingdom classification. Okay, so what is kingdom classification first? You know there are nearly 30 million varieties of living organisms on earth. Okay, they are not similar to each other, they are different from each other. You can see different kind of people. Some are taller, some are shorter, some are fatter. Okay, you can see different types of animals. There are dogs, cats, insects, birds. Okay, they all look different. The all plants look different. The all animals look different. Okay, so there is a lot of variety. There are 30 million varieties of living organisms on earth. Okay, so the scientific way of identifying, grouping and naming these living organisms, that is known as classification. Okay, so it is the universal system of arranging those living organisms based on their similarities and differences. So if anyone will ask you what is classification, it is a scientific way of identifying, naming and grouping living organisms. And why is it necessary? Why is it important to classify? Because it gives us an idea about the evolution of organisms from simple to complex ones. Okay, we can get an idea of how the organisms they they got evoluted from simpler to complex ones. Okay, now what are the bases of classification? First of all, we have to discuss that the branch of biology that deals with the classification of organisms, that is taxonomy. Okay, the branch of biology that deals with the classification of organisms, it is taxonomy, and the scientist who studies about it, that is a taxonomist. Okay, he is a taxonomist. Okay, so now what was the basis of classification? You know, all the living things in earlier classes in 2nd or 3rd you must have studied, all the living things, they were divided into plants and animals. Okay, because they were very different from each other. Okay, animals could move from one place to another for their food or shelter, but plants, they remain fixed in the one position or one place because they can make their own food. Okay, so animals were always dependent on the plants for their food. So on this basis, they were classified into plants and animals on the basis of movement or the mode of nutrition. Okay, now what was the advantage of this advantage of classification? There are 4 points in the advantages of classification. First one is it provides a systematic and simple approach for the study of various organisms. We can study about the different organisms in a very simple way. We can identify new organisms. We can place them in specific groups. Okay, then we can generalize the characteristics of groups so that we can understand all the characteristics of each member of the group. If we are studying about a group, we don't need to study the characteristic of each and every member. Okay, we can just read the characteristics of the complete group and we can know about the characteristic of each member. Okay, then it highlights the relationship between the organisms and give an idea about how organisms evolved from simple to complex ones. So these are the advantages of classification. Okay, now what is the history of classification? How was the animals or the living organisms, they were classified? Okay, so the Greek philosopher, his name was Aristotle. Okay, Aristotle, he was the first one to classify the animals. Okay, and he classified the animals on the very simple basis, on the basis of like presence and absence of red blood cells, or whether the body covering is hard or soft. So on very simple basis it classified the animals. So who classified the animals? Who was the first to classify the animals? It was Aristotle. He was a Greek philosopher. Okay, then his pupil, his student Theophrastus, he was the first to classify the plants on as herbs, shrubs and trees on the basis of their size. So he classified the plants on the basis of their size as herbs, shrubs and trees. He was Theophrastus and he was a pupil of Aristotle. He was a student of Aristotle. Okay. So both the above mentioned systems of classifications, they were known as artificial systems of classification. Okay. Why they were known as artificial system of classification? Because they were based on artificial characters that could be difference in their origin, internal structure or functions. Okay. For example, in case of neem and pine trees, if we are taking example of neem or pine tree, both the trees, they are likely to be placed in the same group because they have this same size. Okay, they are long enough, but both of them have a number of different characteristics. They have different characteristic also. Neem plants, it bears flowers and fruits, but the pine tree does not bear flowers and fruits. So how they can be characterized as in same group? Okay, and in case of animals also, both the birds and insects, they have wings, but they cannot be placed in the same group because really both are different organisms. So there was a lot of confusion. They were simply based on the artificial characters. Okay, they were all artificial system of classification. Then came Linnaeus. So Linnaeus in 1758, he gave a two kingdom classification on the because they classified the plants and they classified the animals. So he gave a two kingdom classification, and that was the most scientific method of classification. He classified all the living organisms into two major groups, and they were Plantae and Animalia. Okay, so Plantae, it consists of all the plants that can prepare their own food, and Animalia, because animals cannot prepare their own food and they depend on the plants directly or indirectly for their food. So he just classified it into two major groups, but there were also problems. So what were the problems? He failed to categorize a few organisms. They were Euglena and viruses. Why? Because Euglena, it is an organism which possesses chlorophyll like plant, but it lacks the cell wall. Therefore, it can neither be grouped under plants nor animals. Now, there are two characteristics in Euglena. Okay, it possesses the chlorophyll, so it can make its own food. Okay, so it should be placed in the Plantae group. Okay, but all the plant cells have the cell wall, but Euglena didn't have a cell wall. So how can we place it in the Plantae group? So he failed to categorize it into the Plantae or the Animalia group. Then next one was viruses. So they are the non-living particles, and they behave like living only when they enter in the living cell, in contact with the living cell. Okay, so he failed to put these two things, Euglena or viruses or a few more organisms, into these two major groups. Okay, so these were the shortcomings of two kingdom classification, and it finally led to the five kingdom classification. Okay, so before studying the five kingdom classification, we will first discuss about this main point here in more to know the different taxonomic levels. Okay, so what are the different taxonomic levels? All the organisms are grouped under the following taxonomic levels. See, first comes the species. Okay, species, there are different species. Then different species, they combine together to form a genus. Different genus, they combine together to form a family. Okay, now different families, they combine together to form the order. Different orders combine together to form the class. Then comes the phylum, and then comes the kingdom. Okay, the whole things, they combine together to form the complete kingdom, plant kingdom, animal kingdom. Okay, so there are different kingdoms. So these were the different taxonomic levels. Now, who was known as father of taxonomy? Because he gave a two kingdom classification and it was the most scientific method of classifying the organisms. So the father of taxonomy is Carolus Linnaeus. Okay, you have to memorize the name Carolus Linnaeus. So after the shortcomings of two kingdom classification, Thomas Whittaker, he was a great scientist. He was an American plant biologist. He proposed a five kingdom classification. Okay, he classified the groups on the basis of five kingdom classification. So there were five kingdoms in this five kingdom classification. They were Monera, Protista, Fungi, Plantae and Animalia. Okay, you have to memorize all the names. They were Monera, Protista, Fungi, Plantae and Animalia, and they were differentiate on the different bases, different characters like cell type, cell wall, nuclear membrane, nucleus, body structure, and mode of nutrition. So these were the criteria of the classification. Okay, so how are they classified? First we will discuss about the criteria of the classification, then we will come back to this table. Okay, so what was the major criteria of the classification? See, criteria of five kingdom classification, it is given on your more to know. Okay, so five kingdom of classification was, first one was complexity of organism. Complexity of organism means they are made up of a single cell or they are made up of many cells. If they were made up of single cell, they were termed as unicellular, and if they were made up of many cells, they were termed as multicellular. Okay, next one was complexity of the cell structure. Complexity of cell structure means they were having a nucleus or not. Okay, so there were two, they were prokaryotic and eukaryotic. In prokaryotic, there was no defined nucleus. Nucleus was not present. Okay, but in eukaryotic, nucleus was present. Okay, now third one was lifestyle. They were the producers, the consumers, or the decomposers. Okay, and last one was mode of nutrition. Mode of nutrition means they were autotrophic. They made the food by themselves, or they were heterotrophic. They depend on others for their food, or they were saprophytic because they decomposed the dead animals and plants. Okay, so these were the criteria of five kingdom classification, and on this basis this table was made. On this basis this table was made. Okay, so these five kingdoms, how were they classified? On the basis of cell type. Cell type means whether they were having the nucleus or they were not having the nucleus. So Monera, only Monera was prokaryotic because in Monera the nuclear material was scattered in the cytoplasm. It was not having a well-defined nucleus. Okay, but in Protista, fungi, Plantae, Animalia, they were all eukaryotic. They were having the nucleus. Okay, now second one was cell wall. Monera is non-cellulosic. Okay, Protista present in some. Cell wall is present in some of the members of Protista. Fungi, cell wall is present but it is without cellulose. Cellulose is not present in that cell wall. Okay. Plantae, cell wall is present with a cellulosic cell wall. There is a cellulosic cell wall also. And in animals, cell wall was not present. Okay. Next came your nuclear membrane. So since there was no nucleus, so nuclear membrane was also absent in Monera. And in all the other kingdoms, nuclear membrane were present because they all were eukaryotic. Nucleus was present, so therefore nuclear membrane was also present. Okay. Now nucleus, we have studied, nucleus is absent, the nuclear material was scattered in the cytoplasm. But in all the other kingdoms, Protista, fungi, Plantae and Animalia, nucleus was present. Therefore they were termed as eukaryotic. Okay. Now body structure, Monera and Protista, they were unicellular, but fungi, Plantae and Animalia, they all were multicellular. Okay. Now mode of nutrition, Monera was a heterotrophic but sometimes autotrophic. Okay, Monera was sometimes heterotrophic, sometimes autotrophic. Protista was heterotrophic, autotrophic also except Euglena. Okay, in fungi, fungi was heterotrophic, Plantae was strictly autotrophic and Animalia were strictly heterotrophic. So these were the five kingdom classification and the differences in the five kingdom classification. The differences or the similarities. Okay. So this five kingdom classification, it was given by Thomas Whittaker in 1969 and it was based on some definite criteria. Understood this much? Now we will discuss about the different kingdoms individually. Okay, so our first kingdom is kingdom Monera. So it had the following characteristics. They were the unicellular organisms. They were only made up of a single cell. Then they did not have a well-defined nucleus. Okay, they were prokaryotic. Okay, then they lacked many other cell organelles also. They were found in higher cells. They have an outer covering called the cell wall. And some of them were autotrophic, while some others were heterotrophic. So these were the main characteristics of the kingdom Monera, which we have discussed in the table also. Okay, now the example of kingdom Monera is bacteria, which is very important. So we will study now about bacteria, the occurrence, the structure, the nutrition, everything about the bacteria. Okay, so bacteria, they are the most primitive and simplest organisms on the earth because they do not have a well-defined nucleus. Okay, first thing, that is the characteristic of kingdom Monera. So they do not have a well-defined nucleus. They are the simplest organisms and they are unicellular. Okay, so bacteria is strictly unicellular and it is prokaryotic. prokaryotic. Some bacteria are autotrophic and some of them are heterotrophic. Okay. Now where does this bacteria occur? So bacteria, they are found in almost all the places wherever the life is possible. It can be in air, it can be in water, it can be in soil, streams, oceans, everywhere, dead and decaying matter, everywhere where a little bit of life is possible, bacteria can be present there. A large number of bacteria, it can occur in our bodies also, human bodies or animal bodies. Okay, they can live both in the presence of oxygen also or in the absence of oxygen also, and they can survive in extreme hot conditions or in extreme cold conditions. So they are very adaptive. Okay, so these were the occurrence of bacteria. Next comes your structure, which is very important. You can look at the diagram given in your book. Okay, so the bacteria have a very simple structure. They have a capsule around the cell membrane. You can look into the diagram. Okay, they have this capsule. You can see the curves. Okay, so this is their nuclear membrane, the cell membrane, and they have a capsule around it. They do not have a proper nucleus because it is prokaryotic, and the nuclear material, see, it is scattered in the cytoplasm and it is known as nucleoid. You have to remember the term nucleoid because the nuclear membrane, the nuclear material, it is scattered in the cytoplasm, so it is known as nucleoid. Okay. Then the cell wall, it is made up of a material that is chitin. You have to underline this word, chitin. Okay, and it encloses the cell membrane of bacterial cell, and its size varies from 0.2 to 100 microns. It can be a very small or it can be very huge. Okay, so it varies from 0.2 to 100 microns. Okay. Now in bacterium cells, ribosomes are also scattered throughout the cytoplasm, and most of the bacteria, they cannot move, they remain still. But some have flagella for their movement. You can see these strips. Okay, you can see these strips. So these are the flagella. Okay, these with this help of this flagella, bacteria can move. Okay, some of the bacteria can move with the special organ that is flagella. Okay. Now comes some other shapes of bacteria. So bacteria are of mainly 4 shapes. You have to memorize the name, that are cocci, bacilli, spirilla and vibrio. Okay, cocci, bacilli, spirilla and vibrio. So first one is cocci. These are spherical in shape. They are true spheres, helmet shaped or kidney shaped. Okay, they are in the shape of kidney. Okay, now they may occur alone, in pairs or in groups. They can occur alone also, they can be in pairs or in groups. If they occur alone, if they occur in pairs, they are known as diplococci, and if they in irregular clusters, irregular groups, then they are known as staphylococci. Okay, and if they are in chain, then they are known as streptococci. Okay, so there are 3 shapes: diplococci, pairs, staphylococci if irregular clusters, and streptococci if they are in chain. So the cocci shaped bacteria, they can cause bacterial pneumonia. Now second one is bacilli. These are rod shaped bacteria. They can be single or they can be formed in chains. They feed on organic materials and they are responsible for the spoilage of food. If your food gets spoiled, that is because of the bacilli bacteria, and they are in the shape of rods. Okay, you can look into the diagram. Now next one is spirilla. These are spirally coiled structures, and there are 2 types of spirillum. Short rods that are slightly bent, okay, they are slightly bent, and the rigid spirilla. So spirilla, they are solitary bacteria and rarely found in chains or clusters. Solitary means they are always found alone, they are rarely found in groups or chains. Okay. Next one is vibrio. These are short, curved or comma shaped. Okay, they are in the comma shaped structures, and acute diarrheal disease called cholera, it is mainly caused by this bacteria, and the bacterium name is Vibrio cholerae. You have to memorize the name, Vibrio cholerae. Okay, so these were the shapes of bacteria: cocci, bacilli, spirilla and vibrio. Okay, now comes useful bacteria. So some bacteria are useful to us also. Okay, some bacteria are useless or some bacteria are useful. So what are the useful bacteria? It is used in the manufacture of curd, cheese and vinegar. So bacteria, they are used in making curd, cheese or vinegar. These are manufactured by which process? Fermentation process. किसी चीज़ को सड़ा के बनाना, that is your fermentation process. Okay, so some in other industrial products such as lactic acid, citric acid, alcohol, they are also obtained by the action of bacteria. Okay, now second one is retting of fibers. Bacteria are used in the retting of plant fibers. What is retting of plant fibers? Due to action of these bacteria, plant fibers, they are separated from the stem. So separating the plant fibers from the stem, that is known as retting of fibers. You have to memorize this term, retting of fibers. Okay. Now next one is tanning of leather. Tanning means when you soften the raw leather or hides obtained from the skin of dead animals. That is your tanning. So it helps in tanning of leather. Then they are also useful in producing certain antibiotics or vitamin B tablets. Then saprophytic bacteria, they are also known as nature's scavengers because they decompose the dead organic matter. Then biogas, some bacteria helps in producing biogas, okay, and it can be used up for cooking or lightening up fuel, okay. And in absence of oxygen, the human waste, cow dung, garbage, they get decomposed by the bacteria, okay, and they can be useful to produce the biogas. Then next one is nitrogen fixation. Some bacteria, they are present in the root nodules of the plants, okay. They absorb the nitrogen and then they convert it into nitrates, okay, so that they can be used by nitrogen deficient plants. Then it helps in decomposing the organic waste or in the sewage disposal plants. Next one is they live in the digestive tract also. So they help in cellulose digestion in herbivores animals. Okay. And last one is curing of tea and coffee. Curing of tea and coffee means to enhance the flavor of tea and coffee. So it is used in curing of tea and coffee also. Okay. Then comes your harmful bacteria. Some bacteria are useful but some are harmful. So some can cause certain disease in human beings like tuberculosis, diphtheria, tetanus, cholera and typhoid. These are the disease caused by harmful bacteria. So today we have discussed about the different 5 kingdom classification and we have started the individual description of the kingdom. So first one was bacteria, okay, kingdom Monera, and the example was bacteria. So this much is enough for the today's video. What is your homework? This is a very tough chapter, so just revise, okay. You have to revise the topics taught, okay. Thank you.